Good afternoon, and welcome to our inaugural Sight and Life DSM Scholar in Residence program and lectureship. My name is Keith West. I direct the program in human nutrition here at the Department of International Health at Johns Hopkins. And we are very excited to have this opportunity to bring Dr. Michael Fenech to Hopkins to be our lecturer all week. 5 lectures to address the very interesting area and emerging area of the role of and our understanding of nutritional genomics in personalized nutrition and in public health. This program is being brought to us through the generosity of a grant to the school by DSM through the Sight and Life Foundation in Basel, Switzerland. It is part of a program where there are scholarship funds available for doctoral students and master's students to pursue micronutrient deficiency prevention in health. DSM and Sight and Life provide technical assistance in our research on micronutrient deficiency prevention around the world through assistance to the technical assistance to our reference laboratory. We have an endowed, or rather a tenure track faculty position that has just been filled by Dr. Amanda Palmer as an assistant professor that is supported through this grant to the school. We have this lectureship. DSM and Sight and Life also provide encouragement and help where possible to accelerate the process of understanding the burden of micronutrient deficiencies throughout the world. And through Sight and Life, we are able to field interns for our Masters of Science in Public Health program to field locations for students to get an in-depth experience at a program level or a research level on this important area, this important problem. This is our 100th anniversary of the school, and so it's auspicious that we are launching this activity during this year. The lectureship was conceived as an opportunity to offer high visibility way to inform our academic community of the public health importance of micronutrient deficiencies and to inspire innovation on the assessment and prevention of hidden hunger. The Scholar in Residence program can provide students, postdoctoral fellows, and faculty a special opportunity to engage leaders in nutrition sciences and explore the collaborations that could occur to advance dietary assessment, micronutrient status assessment, and the prevention of micronutrient deficiencies. So you have in your program a short description of our lecturer this year. Michael Fenech, he leads an internationally recognized nutritional genomics and genetic toxicology research team developing novel gene health assays, including the cytokinesis block micronucleus assay, which is the gold standard for measuring DNA damage, PCR methods for measuring absolute telomere length and telomere base damage, and the cytome assay, which comprises 6 complementary biomarkers of DNA damage and cytotoxicity, adaptable to personalized nutrition, but we also would like to see this technology move forward into public health research more and more. Michael is co-founder of global projects on micronuclei in human populations and leads the Micronutrients Genomics Project. His research focuses on effects of nutritional and other stresses on telomere length, telomere integrity, and personalized nutrition approaches to prevent dementia, cancer, and other chronic diseases. He is a recipient of numerous awards, including the Alexander Hollander Award in the United States, and holds honorary professorial titles at universities of South Australia and University of Adelaide, Flinders University, and Taipei Medical University. He's co-authored over 300 papers averaging nearly 50 citations per paper. He's really a leader in this field, and he has a very simple aim in life, to determine levels of dietary intake, and nutritional adequacy needed to prevent DNA damage and promote healthy aging. And as one gets older, the more important that topic looks. Our 5 lectures this week deal with nutritional genomics and the sort of a background, history, interactions, a systems biology approach for incorporating nutritional genomics into our research, nutritional developmental determinants of health and disease mechanisms and prevention, and then how we wrap this into a public health and personalized nutrition approach. The contents of the lectures. Just Klaus could not be with us today, but he will be joining us tomorrow. But just on behalf of Sight and Life Foundation and DSM, he had these few words: that Sight and Life Foundation and its parent organization, DSM, are pleased to support this Scholar in Residence program at Johns Hopkins. The program seeks to provide students, fellows, and faculty with unique opportunities to engage global leaders who are advancing our understanding of micronutrients and dietary quality to improve public health. Our aim is to promote academic exchange and stimulate collaborative research to improve diets and prevent malnutrition. As the inaugural Scholar in Residence, we hope that a week with Dr. Fenech on campus will generate insights that enable scientists and practitioners to apply the new biology of nutrition to shape nutrition programs in the future. Michael, it's a pleasure to have you.
Thank you. Thank you very much, Keith, for the generous introduction, very informative introduction. And the why not brings me here and gives me this opportunity to try and extend myself. And I wonder, as I go along to these lectures, keep on asking myself, have I bitten more than I can chew? And so I'll do my best, but if I fall short every now and then, I do apologize. So the whole concept here is what is the interaction between what we put into our mouths and our genome? If you had to pile up all the food you eat in a lifetime, it would be quite a pile, very messy. And out of that, our body seems to make some sense that allows us to grow and be healthy, reproduce. The problem is we don't do a very good job of our choices from what we've learned in the past and from what we are doing now. And the hope is that nutritional genomics and our understanding of nutrient-gene interactions might help us to move forward. Now, the purpose of this particular lecture, the first one, is to cover some key points regarding the evolution of the science of nutritional genomics, trying to distinguish between the 2 key aspects of this field, which is the nutrigenomics aspect, the impact of nutrition on the genes, and the genetic aspect that determines to some degree what happens in that interaction. The role of genetic variation in nutrient metabolism and food preferences, and the effects of nutrients on dietary patterns on gene expression. And this last bit will actually be very brief because I'm really looking at it from the historical aspect, something about the very first studies that were done in this field that I'm aware of, and so on. Okay, so this is my brief history of nutritional genomics on one slide. It's not a very well informed slide. I didn't have a lot of time to go into deepest parts of the history and the detailed, and there are so many characters involved, you couldn't possibly do justice. But it just gives you a bit of a flavor. Well, before we knew much about science, well, humans were always in a way inquisitive. That's why we're here today. And we were doing experiments, trying this and trying that. So in the early days, thousands of years ago, when the first hominids came on the scene, there was a lot of discovery of healthy and unhealthy foods by trial and error. And our tongue, in some ways, and the way the food tasted, helped us along. and what we observed. Eventually humans migrated out of Africa, and there was more adaptation to be done with the new diverse climates and environments that they encountered. And the shift from hunter-gatherer to agricultural living in itself created more science that humans had to develop in terms of breeding plants and animals that could be cultivated and or grown or bred for particular traits, right? So, and that has a lot to do with changing also the dietary pattern towards a diet based on grains, on dairy, and so on. But one can't talk about the history of nutrition without mentioning Hippocrates, because conceptually, well, he put together this idea that food should be seen also as medicine, and that the choice of foods therefore can affect our health, and that is a very powerful concept and message that still focuses our minds with respect to that. And he clearly explained this very well and put it into practice in his own health spa, the Escapalion, on the island of Kos in Greece, which if you haven't been, I recommend visiting just to get a sense of the person and the place. The fact that macronutrients on their own would not be enough to maintain health became evident in the late 19th century, and the concept and the discovery of vitamins, here showing Howard Hopkins as one of the pioneers in this field, key pioneer in this field, and is it Ed Blundell, I think, here, and Hopkins, pioneering the work relating to vitamin A and D, and others showing the way with regards to vitamins and minerals and their importance, and ultimately the discovery also of inborn errors of metabolism, such as phenylketonuria, and the discovery of DNA itself, which then led to all the technologies and possibilities that enabled us to understand genotype in great detail, and also gene expression, which resulted gene expression analysis, which is one of the very first fields in which the impact of nutrition on the genome and its expression was enabled. And of course, now we live in the field and the exciting and mind-boggling era of multi-omics, where we can study the impact of nutrition and, in fact, any environmental factor in many different, using many different omic technologies, and the vast amount of data generated and its interpretation is really going to be a huge challenge. So we are at a very early stage of evolution when we have many bits, many thousands, hundreds of thousands and millions of bits of information that we need to put together in a way that is usable and practical. And that's really where we are, and the history is still being written. So to try and understand a bit about human nutrition, we kind of need to go to the beginning. In that there was quite a big change when in human diet it is taught, when humans started to know how to use tools to process food or to find food. There's a very big difference between, let's say, the diet of a chimpanzee and what a human eats. And I haven't had time to show this slide, but experiments have been done where the diet of a chimpanzee as opposed to a human diet was fed to mice, and this resulted in great differences in gene expression. But interestingly, the genes that were affected were also the ones that were mutating a lot during the evolutionary process. So there may be something to do between the gene expression effects and the mutations that we eventually accumulate as an adaptation. A very important aspect, of course, regarding this ability of using tools which enabled humans either to kill animals or cut up the flesh of the animals, to dig out tubers and maybe to fish and so on, is thought to have had a very important impact on encephalization, or the growth of the brain, or the evolution of a bigger brain and a more complex brain. But this is really a matter of hot debate. To what extent, which food made a difference, we're not so sure. But what is known is that over the millions of years and the past 200,000 years, there has been a large increase in cranial capacity. A lot is thought that this probably has maybe more to do with the consumption of aquatic animals such as fish, maybe something to do with the omega-3 fatty acids, but that's probably only part of the story. But clearly, humans are on a very different trajectory, and our trajectory and our foods are becoming even more complex and diverse because of processing. Whilst at the same time, the diversity of foods in terms of what we buy in the supermarket is obviously a lot less than what is available in the wild. So the reasons why humans moved out of Africa is not so clear, but a recent paper published in Nature suggests that this was maybe to a large extent driven by changes in climate change, which enabled the passage out of Africa through the southern West Asia, maybe because that part of the land became habitable again, and this allowed a number of waves of humans to get to migrate into Eurasia and eventually to the Americas and Australia. During this process, there was quite divergence in the genetics of the human population. And there are distinct genetic variants distinct to the African populations, Europeans, Indians, and East Asians. Intriguingly, the genetic diversity in the African populations is actually, in the diverse African populations, tends to be quite larger than is in fact, let's say, in Asia or in Europe. And these diversities are evident in the polymorphisms that one can see and measure in the different populations. Now, with the migration of humans, there's also the adoption of the agrarian way of life and different lifestyles depending on pasture, use of the shift from hunter-gatherer to the use of pasture for beef and dairy, let's say, and cropland for grains and vegetable growing. And over the thousands of years, it is reasonable to expect that humans evolved to or adapted to those to a certain degree to these new ways of eating and being. But in many ways we are still maladapted even to these ways of being and living. So I like to show this slide because this shows us where human migration has taken us and how we have evolved. So if you look at what a family in different parts of the world eats today, there's a huge difference between, let's say, what is eaten here in the USA, in the developed countries, Germany, where there's a large amount of food eaten, a lot of calories, a lot of processed food, as opposed, let's say, to Chad and Africa, where you can see with a minimum set of foods, one can still thrive and be and reproduce to different extents of health. And you can see also the proportion of plant food to dairy or meats and so on is really very diverse. So the question is, there's a great diversity here in the exposomes. In this case, this is a word that's used maybe too easily. It refers to the exposures, which includes nutrition, but also environmental. It could be the climate as well. It could relate to stress as well, which is different in, let's say, a city as opposed to living in the countryside. But all of these interact in many different ways. And the nutriomes, or the nutrient combinations that one ingests, and the interaction of these with the genome and their effect on the epigenome, which we'll talk about tomorrow in more detail, and their interaction. Now, so in many ways, I usually don't give this talk in this order. I usually talk about nutrigenomics first before I go to nutrigenetics. But because we are talking about the genetic aspects, I thought, well, let's try the other way and see how it goes. So what do we actually mean by nutrigenetics? This slide, which I've kind of adapted from Martin Kussman's slide in this special issue of Nature Outlook in 2010 on nutrigenomics, tries to capture what we really mean by nutrigenetics. I also call it the ethics and the omics for personalized nutrition, because in a way there are aspects of human ethics that come into it when we use this kind of information. Now, we really use genetics in this case to understand predisposition. So does the individual have a good predisposition to absorb this nutrient or not? To absorb it into the bloodstream and into the organs, and to metabolize it appropriately, and so on. So that's what we mean. That's why we would like to know about genetics, because that can tell us whether an individual has a particular difficulty to utilize certain nutrients and metabolize them in a proper way. Epigenetics is kind of a different aspect of genetics, and that is really to know and understand which genes are being expressed and why that expression has altered. And this could relate to marks, methylation marks on DNA or on chromatin. It could be acetylation. It could be other kinds of marks on the chromatin that determine the extent to which a particular gene is expressed or not, or that is accessible for nutrient factors to actually activate certain genes. So really we want to know epigenetics to understand programming. So we want to know how the genome is actually activated and working in the individual. Ideally, what we would like is to use this information so that we can organize or put together some personalized nutritional intervention that we think will be more efficacious for that individual or genetic subgroup. But we need tools to know whether the recommendations we are giving are actually causing benefit and not harm. Very important to do that. And one could therefore use the new sciences, omic sciences, let's say of transcriptomics and proteomics, to measure the molecular response. And that is very important because that infers whether one is going in the right direction or not. Admittedly, right now we do not have enough information to be able to use this to know how to use this knowledge very well as yet. We're improving all the time. We're getting there. It's getting better all the time. That we can at least know what a profile, what a transcriptomic or proteomic profile is consistent with improving health rather than not. And this may be different depending on which aspect of health one is looking at. But the differences between these two and metabolomics and genomics is that metabolomics and genomics gives you another aspect in that the metabolome is kind of the sum total of what is happening in the cells at the molecular level in terms of how the molecular transactions of life are being performed and whether they're being performed efficiently or not. So the metabolome will inform us about the efficacy of the intervention, exactly in terms of how the nutrients and the metabolites are transiting and shuttling between organelles and within tissues. And within tissues. And the genomic side is really important because it really tells us about a specific aspect of the metabolome, which is DNA metabolism. And that is whether the choices made are actually not causing harm to the genome, right? Or enabling the genome to replicate itself properly. Because ultimately, we are here because our cells have divided. If I look at the buccal mucosa, the cells I have now are kind of photocopies of what I had 21 days ago. They've changed over. If you look yourself in the mirror and you think you're looking a bit different, it's because the cells have turned over. That's all that's happened since we were born as a single cell. It's just one copy of a copy of a copy of a copy. And the ability to live healthier and longer depends a hell of a lot on the regenerative capacity of our tissues and the ability to regenerate properly and appropriately. Okay, so what about the human genome? So we know a hell of a lot, and there are many aspects to this. Of course, there's the nuclear genome, which is the bulk of the human genome, and there's also other genomes, such as the genome within our mitochondria, which we inherit from our mother. And each cell has got many mitochondria, and each mitochondrion has got many mitochondrial genomes. There's a big difference in size. The nuclear genome is 6 billion base pairs. The mitochondrial genome is about 16,600 base pairs. The nuclear genome is distributed in 2 by 22 autosome chromosomes, and either 2 X chromosomes or X and Y, depending whether you're male or female. But 98% of the nuclear genome does not code for proteins. However, 80.4% is thought has some functionality, probably regulatory. So all that DNA that was thought to be extra is actually very important in regulating how the genome is expressed in the end, with a lot of levels of management, so to speak, of how that expression occurs. 2% of the nuclear genome is coding sequence for proteins, whilst in the mitochondrial genome, probably about 90% is coding sequence for proteins. So the mitochondrial genome is very, very tightly packed with information. And the mitochondrion itself needs proteins that are encoded in the nuclear genome that are transported into the mitochondria also to function. So there are about 20,600 protein coding genes, about 34,000 novel non-coding transcripts according to the latest versions of ENCODE and GENCODE, and about 9,600 long non-coding RNA loci. And there's about 11,000 so-called pseudogenes. These are versions of genes that are still hanging around, which are do not function well. However, despite all of this, the inter-individual difference between humans is relatively small, about 0.1%. Between human and chimpanzee is just 4%. So the expression of genes really makes a big difference to these differences that we see between people. During this series of lectures, I will not be talking about a very important aspect of nutritional genomics that is emerging, because there simply is no time, and you could spend 5 lectures just talking about this topic. And this is the microbiome. These are the bacteria that live in our gut. We know that they have a strong impact on health. I'll mention it briefly in one of the lectures. But this is a whole new and emerging field and could make a very important topic, let's say, for a series of these lectures, maybe next time around. But this is a really also very exciting field. Now, what types of variations are present in the human genome sequence? Well, you can have single nucleotide variants. You can have an insertion-deletion variant. It's called so because you're not sure whether this gap happened because there was a deletion or because GAT was inserted, right? So you can't really tell that. That's why it's called insertion-deletion. You might have a block substitution here. So this sequence here, AGTG, wasn't substituted instead of CCCC, or an inversion occurring in part of the sequence, or you can have copy number variations, and all of these can occur to different degrees. So you can get the single nucleotide polymorphism is the most frequent. So there's about 3 million to 4 million of those. Here you can see the number for Craig Ventnor, who was director of the Human Genome Project, and James Watson, one of the discoverers of DNA. Very similar. They're both of European origin. What is noted here, this is the genome, the SNPs in a person of African origin. There tends to be a higher frequency of polymorphisms in people of African origin. It's in some studies, but not all, necessarily all studies. If you look at the variation or the incidence, clearly SNPs are the most frequent. They're in the millions. Then you've got block substitutions, about 53,000, indels, about 851,000, inversions, only about 90 on average, and copy number variants, even less, 62. But they're still important, and I'll show you some examples. Now, we talk about nutrigenetics as if it was something that only happened yesterday, but in fact, we became aware of the genetic aspects of this of nutrition almost 90 years ago or 100 years ago, and this is I'm referring here to phenylketonuria, which is one of the first inborn errors of metabolism that were discovered, and it was evident in those days. Ashbourne Falling in 1934 first discovered phenylketonuria after testing the urine of two mentally challenged siblings, and he isolated phenylpyruvic acid in the urine in these individuals. It turns out that this is due to a mutation. There's about 500 different kinds of mutations that might affect phenylketonuria in the phenylalanine hydroxylase. This non-functional enzyme results in the accumulation of phenylalanine and phenylpyruvate and other metabolites that harm the brain and cause intellectual disability. And the gene happens to occur here on chromosome 12 on the Q arm. Here is some results from the urine tests done by others, such as Penrose and Questel, showing here the high level of phenylpyruvate in the urine of a case compared to a control. Eventually it became evident one way to manage this disease is to restrict the intake of foods rich in phenylalanine, and these are foods, mainly flesh foods like pork, chops, beef, cheese, dairy foods, and so on, chicken meats, and so on. All those foods we eat in abundance today. So there are ways. So here we have already a dietary solution to an inborn metabolic disease, which was discovered almost 90 years ago. So the concept of targeting nutrition is not new. There are at least 80 such inborn errors of metabolism that are tested in newborn screening programs. And this is just some other examples. Here are some examples in fatty acid oxidation disorders relating to genetic defects in mitochondrial metabolism. Here is a defect relating to vitamin B12 metabolism because when B12 is defective, when methylmalonyl-CoA mutase is defective, which requires B12, then methylmalonic acid accumulates, and so on. So this is not new. What is new is that we are expanding this science to more common mutations which have more subtle effects on health. And this broadens the application of this field massively than to just 80 inborn errors of metabolism. Of course, these are important, and we may in fact acquire these kinds of phenotypes as we get older and as we accumulate more mutations, or perhaps because these genes are switched off epigenetically due to the epigenetic drift that occurs with aging. Except they are most evident, and the science is applied here in during baby screening programs. Now, I was asked to say something about taste. It's not a field I know a hell of a lot about, but it's very important because it determines to a large degree what foods we choose to avoid or we choose to eat. And of course, the food industry invests a lot of energy and money in this field because it helps them find out how to put foods together to make you buy their food more often. This is big business, huge business. And therefore, the genetics and science of this is very important economically. It might not be so good for our health because it can be manipulated in a way that takes us to eat the wrong foods rather than the ones that benefit us in the end. But of course, there are many aspects to this. It helped, of course, in thousands of years ago, millions of years ago, it helped our ancestors to distinguish between foods that may not be safe to eat by their bitterness, let's say. And of course, it helped appreciate other foods based on whether they were sour, salty, or sweet to eat. And obviously, we are very well primed to and affected by these experiences. But this kind of information can be used here, for example, to know what way to put together, let's say, a sandwich or a hamburger so that your customers are more likely to buy it depending on their genotype. And here, for example, we can see these are the various chemicals associated with these foods, such as sucrose in relation to onion, glutamate in the case of tomato, isothiocyanates, thiocyanates in the case of watercress, isovaleric in the case of cheese, and aldosterone in the case of ham, which relates to the boar taint. Now, this particular person happens to have the genotypes that would tick the box for them. Why is this one not? And hence, the liking scale is obviously going to be quite different. So this is really a commercial way or application of understanding of why you might choose certain foods or combinations as opposed to others. Of course, we do this subconsciously. But I'm going to go back now to the population kind of studies. I apologize if I jump a bit in and out like this, but maybe it makes it a bit more varied. It also helps to bring it back again to what we were talking about. I just wanted to give an example here of copy number variation, which I referred to earlier, which is not so common in terms of the mutations you see, but is relevant in terms of the adaptation of humans to different diets. What is observed here is that the gene for amylase can occur in different copy numbers, anything between 4 to 8. And what's observed is that in those populations which have got traditionally a diet rich in starch, tend to have a higher copy number variation of amylase, which enables them to digest the starch that they are eating, whilst the ones with lower starch diets tend to have lower copy number variation. So this appears to be an adaptation, perhaps, to these new diets. Now, it's also observed is that the number of variants you have of this gene also alters your susceptibility to calorie-rich diets as well. And this can affect the risk of obesity. So it has implications not only to our past and our adaptation to it, but also our maladaptation to current dietary patterns. Another interesting aspect is as a result of the agrarian way of life, of course, we started consuming a lot more milk and dairy and so on. And as a consequence, some adaptation did happen. Now, the lactase gene usually diminishes during childhood due to reduced expression of the lactase gene. So lactose intolerance might be expected in adulthood unless there is persistence of lactase occurring into adulthood. And this might have evolved with dairying around 7,500 years ago. Now, this map shows you the prevalence of lactose intolerance. So the darker the orange is, the more lactose intolerance there is. So you can see in China there's a very high level of lactose intolerance. Now, whether that's because the agrarian way of living there did not introduce the production of dairy, maybe that was an adaptation, or maybe the population itself already had a strong genetic lactose intolerance and therefore never adopted dairying as a way of life. We don't know which way around it was. But it does inform why lactose intolerance is still very prevalent when you think in terms of numbers, because even in these large areas with the lighter orange, we're talking only about 50% who may be lactose tolerant. Yeah. So it's still a very high frequency despite all the thousands of years of living this agrarian way of being. So we still have not really adapted to this. And maybe there are limits to adaptation. I think this is what we need to, this is what my thoughts are, that there are some limits to adaptation. We cannot just push the system, just like we cannot eat as many calories as we like and expect that we are going to evolve quickly to this new way of being. Maybe there are limits, and we know that there are because you start with high-calorie environments, you start getting ectopic deposition of fat, not in the adipose tissue, but in other tissues, which creates disease. Now, the question of lactase persistence or or lactose tolerance has to do also with the origin of haplotypes. This is another genetic aspect which I did not mention. And a haplotype is a group of genes in an organism that are inherited together from a single parent. And the reason being is that these genes are very close together. And you tend to get the same kind of mutations clustering together when you have these genes together. Now, this may relate to health, it may relate to disease. For example, in cancer, an aspect which I would have liked to talk about, because we have a lot of cancer survivors and we need to know what to feed them so that they don't get their cancer back. But a common mutation there is the P16 deletion, which happens to be next to the AMTAP gene needed for methionine metabolism. So these are real issues in terms of how to handle it. Now, in the case of, you can also have haplotype blocks here in relation to mitochondrial DNA, and that's used to track human evolution, migration. Now, in the case of lactase persistence, there is a haplotype here involving the MCM6 that has got certain mutations that are always linked. The MCM6 gene is very close to the lactase gene. And these mutations here in MCM6 and these other point mutations here are associated with lactase tolerance. The mechanism is not known, but there is a number of haplotypes here, A, B, C, and U, which relate to this. And this might explain why people can sometimes tolerate milk in their later life. Another part of the agrarian kind of impact is of course, the high intake of grain, of wheat, which may be associated with celiac disease because of the high level of gluten. Gluten can bind to other proteins in the body, which are normal proteins, and make them appear like foreign proteins, and maybe induce a autoimmune kind of response and inflammation. And this is the condition here. And here genetic variants, for example, for the HLA DQ2 or DQA genes, tend to have a greater propensity to this condition of bowel inflammation. But again, we have some solutions to that with gluten-free foods in the marketplace. Now, I'll just give some other examples that are more pertinent, let's say, to the question of obesity. There are a number of genetic variants that tend to predispose people to obesity. There's a hell of a lot of them. And again, you could do a 5-lecture series just on this aspect of health. But one that comes up very often is the FTO variant. And it appears that people who have this kind of mutation, the RS9939609, tend to have a predisposition or propensity to select foods that are rich in calories when given a choice. And the reason for that is not so clear why that happens. Okay, but the mechanism for this is still being elucidated. But it obviously something's happening in the brain that lights up your brain when you see foods that are sweet or fatty in these people that makes them go for that food more often than not. There are also a lot of interest in the response of people to weight loss diets. It's very hard to have, not everybody succeeds on weight loss diets, right? And some of them might succeed and not, and then flip back to their previous weights that they had before the intervention. Now, Jose Ordovas and his group do a lot of work on this aspect of nutrigenetics in identifying genes that may make it harder for people to succeed on weight loss interventions. And here they've identified a number of mutations in the clock genes which have to do with your diurnal clock in responding to waking up, sleeping well, and so on. And there are mutations in these genes which for some people makes it harder for them to maintain a routine, to sleep appropriately and long enough and at the right time. And these aspects are very important in the success of being able to change a lifestyle and maintain it over time. When we talk of food, we don't, in the Western world, we also have to think of alcohol. Some people might even think that alcohol is a food or a fuel, but it depends on which time of the day you're talking about, and it certainly helps de-stressing some of us. But there are also serious concerns about alcoholism, because we know it's a risk factor for many diseases. And for example, in females, for breast cancer in particular, even moderate drinking of alcohol is an issue. But this issue may be further more important if you happen to have a mutation in acetaldehyde dehydrogenase, which is a fairly common mutation, particularly in Asians. And that is because alcohol is converted very quickly to acetaldehyde in the body through alcohol dehydrogenase. Acetaldehyde is a carcinogen. It's a carcinogen because it damages DNA. It binds to DNA and damages it. And if you're a slow metabolizer of acetaldehyde, you're going to accumulate it more so in the body. And especially if you are binge drinking, then the concentration of acetaldehyde will reach to a level where it's going to harm your DNA seriously. And we can see this very clearly with one of you can do this with a number of assays. This is the cytokinesis block micronucleus assay, which Keith mentioned in the introduction. It's an assay I developed many years ago. It's actually originally developed to study the effect of ionizing radiation. It's actually a technique used to study for biodosimetry when there is a radiation accident. But when I switched fields to nutrition, well, I haven't really switched fields. I've still got a foot in radiation biology and 3 feet in nutrition. This one can be used to study the effects of nutrition and other toxins that we ingest. And you can see here that the susceptibility to DNA damage is higher in slow metabolizers than in fast metabolizers of acetaldehyde. Now, there are many. What about the vitamins? Well, there are many vitamins, and there are many polymorphisms. And I can't talk about all of these, so I'll just talk about one of them here, MTHFR. MTHFR is the methylene tetrahydrofolate reductase enzyme. And the gene, there's a gene with a common polymorphism, C677T. It's the most studied polymorphism. There are thousands of papers, nutrigenomic, nutrigenetic papers on this. There's 250 meta-analyses on this epidemiological studies. So it's massive, it's the most studied. It requires vitamin B2 or riboflavin as the cofactor. And what it does, it converts this form of folate, 5,10-methylene tetrahydrofolate, to 5-meta-tetrahydrofolate, the form you need to convert homocysteine to methionine. So when this enzyme doesn't work well, you don't produce enough 5-meta-tetrahydrofolate, and therefore you don't convert homocysteine to methionine efficiently, and you have high homocysteine, a risk factor for cardiovascular disease, dementia, and also neural tube defects, which I'll come to in a moment. So what I'll be showing is some original papers. So this is the first paper that identified this polymorphism in human cells. And basically what they did these are in vitro experiments showing the high increase in homocysteine. The mutation is actually a thermolabile variant. So in other words, when the temperature goes up, then this becomes more sensitive. Here is a population frequency of this polymorphism. This is the homozygotes of MTHFR C677T around the world. And you can see there's a high prevalence of homozygotes, almost 30 to 35 in China, Mexico, and in I think it's Venezuela, and Italy. And if you look further here, you can see many more polymorphisms in this pathway. You can see here, if we look at MTHFR, C677T, it's low, very low in Africa compared to Europe and Asia. If we take here the reduced folate carrier that brings folate into the cells, this polymorphism which affects this enzyme is very high in Europe, but low in Africa. And these events have something to do with the need of folate to protect against the damaging effects of sunlight, because folate actually protects against UV damage. And this is the reason why it's thought that these polymorphisms are lower, let's say, in Africa than they would be in Europe. There are also other polymorphisms like transcobalamin 2 that transports B12. You can see it's very low here, this polymorphism. Again, you need B12 for proper metabolism of folate, and these may be related. So, you can dig deeper here. You can now, with all this information we have in genomics-wise, we can identify mutations in all of the genes involved in the transport of B12 and folate, and mutations such as cubulin in the case of B12 occurring being associated strongly with B12 status and with homocysteine status. And the same with choline. There are important mutations such as in the PEMT gene, which is required to which is required to generate choline. Now this mutation is also responsive to estrogen. So during when in premenopausal years in females, estrogen activates this enzyme, and the reason has to do with choline being very important for the health of the baby, particularly brain development, because it affects the number of stem cells in the hippocampus and therefore the ability of the brain to develop and grow properly. Very interestingly, the frequency of this particular mutation varies greatly around the world. So one recommendation that you might give in one part of the world will not work in another, depending. So this is why this knowledge is important. Now, we also know nutrigenetics is important because it relates to how you observe things in populations. So you might see a small difference in the effect of a genotype on a health outcome, but that small difference may be occurring only in those with a particular genotype who are practicing a particular diet and not others. So what you see may be dampened by nutrigenetic effects, or it may be amplified if you look in the right place. You might also see no effect because the effect of a particular exposure, let's say diet 2 as opposed to diet 1, goes in opposite directions depending on the genotype. And these effects are seen, in fact, in some studies. The next slide shows you an example here in the Boston Puerto Rican health study in relation to this mutation in MET1A and the interaction with vitamin B6 and the impact on 8-hydroxydeoxyguanosine, which is a DNA damage event caused by oxidation of guanine, which you can measure in urine. And what you're seeing here is an opposing effect depending on the vitamin B level, B6 level, and the genotype. So again, this knowledge does help us to interpret the data that we see epidemiologically and informs intervention. Now, very quickly about nutrigenomics. These are just the last four slides. When we talk about nutrigenomics as opposed to nutrigenetics, we are still using the same kind of knowledge, but in a different way. So we are studying genetics to understand the blueprint, right? So what does this individual's genetics look like? And we are using epigenetics to understand what has been programmed. So it tells us, are they expressing MTHFR or not? Are they expressing PEMT? Is it interacting with estrogen? Is the mutation affecting that interaction in some way? And are those genes, well, later in life, in postmenopausal years, is PEMT switched off because you were eating a particular diet or because you were exposing yourselves to some environmental factor that for some reason causes damage close to the PEMT gene and causes heterochromatic changes in that region and switches it off and makes it inaccessible. Now, in this case, in nutrigenomics, we're using transcriptomics and proteomics to understand what appears to happen, and then metabolomics and genome stability to understand what has happened, and whether in fact there has been any damage done to the genome as a consequence. Because it's not just about gene expression, it's also about understanding how serious, what level of damage has been induced. Because we know the DNA damage rate is a very important and strong predictor of developmental risk of developmental disease for the fetus, and also of the risk of degenerative diseases. And that makes sense because once the genome of a cell is damaged, the ability to replicate is diminished. The ability to make new cells is diminished. And the kinds of cells that are produced often don't look anything like human. And in fact, if you look carefully at some of the karyotypes in older people, you wonder, a chimpanzee would look a hell of a lot more normal than the cell you are looking at. So there are big changes occurring in the genome with aging. And a lot of that has to do with with poor nutrition. Okay, so now we know that the genome can be harmed very early in life. This is the first study that I could find about the effect of malnutrition on chromosome damage. This is in children with severe protein-calorie malnutrition, exhibiting symptoms and phenotypes of kwashiorkor or marasmus. What this study showed was these children's chromosome damage rate was 5 times higher than normal. Now, if I was a radiation biologist and I didn't know that they had protein-calorie malnutrition, I would have thought they have been exposed to quite a serious level of ionizing radiation if I looked at that, because chromosome damage is one of the main biomarkers used to study radiation exposure. And I would say these children are being exposed to levels of radiation that would be considered unsafe. So the key point here, and I'll show you another slide later, is that malnutrition causes as much damage as radiation. Okay? So if we are worried about radiation, we should be a hell of a lot worried about the impacts of malnutrition on damage to the genome. Now we know that malnutrition can also affect DNA methylation, as shown here by this first study on folate. This is a depletion study showing the effect of depletion on homocysteine and increased hypomethylation. So this is a methylation acceptance study. So the DNA was depleted of methylation and can accept methyl groups. That's how the assay was done. This is the first study showing the impact of adiposity, obesity, and diabetes on gene expression in the old days when you had to do and build your own gene expression arrays, for those of us who remember those days, many moons ago. So that was very interesting, and the changes observed were consistent with dedifferentiation of adipose tissue and diminished mitochondrial effects. Again, similar kinds of studies in relation to weight loss, who succeeded, who didn't. This was one of the first studies looking at the changes in gene expression. And finally, studies here with different dietary pattern, the PREDIMED trial, where one of the very first studies where they looked at differences in gene expression affecting pathways relating to atherosclerosis and so on. So the take-home messages are quite simple in a way. That human evolution has resulted in great diversity in human genomes and human dietary patterns. Changes in nutrition can affect genome integrity, gene expression, and metabolism. Nutrigenomic studies may explain why and how malnutrition affects health. Deleterious mutations in genes involved in uptake and metabolism of nutrients are a cause of human non-communicable diseases, and they may be actionable in some cases. Therefore, suitable matching of diet with genotype can contribute to the prevention of non-communicable metabolic diseases. Thank you, and I apologize for going longer than I should have.